Now we'll start with carbohydrates. To know about the carbohydrates, you should know that they are classified into 3 to 4 types: monosaccharides, disaccharides, oligosaccharides and polysaccharides. So monosaccharides, they are found to be simple sugars. So all of you know that they are simple sugars. Example will be glucose and fructose. Disaccharides, they are made up of 2 monosaccharides. Example will be sucrose, maltose and lactose. And oligosaccharide, they are made up of to 9 to 10 carbohydrate units. And polysaccharides, they are made up of more than 10 monomers, and they are formed to have a disaccharide unit and a monosaccharide unit. Now the polysaccharides are of 2 types: homopolysaccharide and heteropolysaccharide. So example of a polysaccharide, all of you know starch is a common polysaccharide in plants, then cellulose and glycogen. Now coming to what are monosaccharides. Monosaccharides have a general formula CnH2ON. They have both open chain structure and ring structures. So they are mostly seen as 6 carbon aldose sugars with 14 asymmetric carbons in their structure. Now the monosaccharides can be classified into 3 or 4 different types based on their carbons. They are found to be called with 3 carbon as triosis, 4 carbon is tetrosis, 5 is pentosis and 6 carbon they are called as hexosis. Now the monosaccharides are found to have at least one carbonyl group in the structure, which is C double bond O. And if they contain an aldehyde group, you call them as aldose, and if they contain a ketone group, you call them as a ketose sugar. Now coming to the D and L forms of carbohydrates. So they are found to be called as stereoisomers. So the D form is said to be dextrorotatory and L form is said to be levorotatory isomers of their structure. Now coming to the different types of properties of chemical reactions or chemical properties of carbohydrates. So first, if you want to see, the first property is going to be seen as mutarotation. So mutarotation is seen between the 2 forms, which is alpha form and beta form, where they are called as anomers. So the alpha anomer and the beta anomer, which is seen to exist in solution. So mutarotation is identified as the difference in the specific rotation of the plane polarized light due to the change in the equilibrium between the 2 anomers, that is alpha anomer and beta anomer, in the particular reaction in the solution. Now if you see here, alpha D glucose and beta D glucose have their own specific rotation, mutarotation values, and they are found to be at equilibrium at the value of plus 52.7 degrees. Now coming to the chemical reactions of monosaccharides. So first we'll look upon the first reaction, action of base on sugars, where the monosaccharides, they react with weak acids and they form salts with high pH. So the first salt which is seen is called 1,2-enediol form, which is seen the reaction with bases. Now coming to the reaction of bases on sugars, so all of you would have heard about non-reducing sugars. So non-reducing sugars and reducing sugars. So sugars which have the free aldehyde group, you call them as reducing sugars, and sugars without the free aldehyde group, where the aldehyde group is involved in bonding with other functional groups. So those sugars with the aldehyde groups which is bonded with other functional group, you call them as non-reducing sugars. Okay, so the main reaction to identify whether a given sugar is reducing or non-reducing, we perform the very important test which is called the Felling's test, Benedict's test and Barforth's test. So these 3 tests tell us whether the given sugar is reducing or non-reducing. If the given sugar is reducing, it turns reddish orange precipitate, and if it is a non-reducing, the color does not change and it remains the same. Now coming to the reduction reactions of carbohydrates. So carbohydrates undergo reduction reactions to produce sugar alcohols. An example of a sugar alcohol is sorbitol and mannitol. Now if you see the further chemical reactions, the very important reaction to identify the carbohydrate is oxazone structure. So oxazones are formed when the monosaccharides react with phenylhydrazine in acetic acid solution to form crystalline structures. So every sugar molecule has its own sugar crystalline structure. So if you see for glucose, it has got a needle shaped structure, maltose has got a flower shaped structure, and lactose has got a powder puff shaped structure. So the different types of crystal structures are seen with monosaccharides when they react with phenylhydrazine. Now coming to the next one, glycosidic bonds. The glycosidic bonds are formed between two monosaccharides to form a disaccharide. So all of you know what is a disaccharide. Disaccharide is formed with a glycosidic linkage. So there are three main types of disaccharides which are seen: maltose, lactose and sucrose. Maltose is composed of glucose units, lactose is composed of D-glucose and galactose units, and sucrose is composed of glucose and fructose units. Now coming to the next classification of carbohydrate, polysaccharides. Polysaccharides are classified into homoglycans and heteroglycans. So homoglycans example are starch, cellulose, glycogen, and heteroglycans are gums and mucopolysaccharides. So coming to starch, starch is made up of alpha amylose and amylopectin, and starch is digested by the specific enzyme called alpha amylase, where alpha amylase will degrade the alpha 1,4 linkage. Now coming to the other classification of polysaccharides, glycogen. So all of you know what is glycogen. It is a very important carbohydrate where it is called as animal starch. So animal starch or the glycogen, it is an important form of storage of glucose in the liver. So whenever you take in glucose in the diet, it is first converted into glycogen, which is the first priority, where glucose is converted to glycogen and stored in the liver. Now coming to the next polysaccharide, which you call them as heteropolysaccharide, where the heteropolysaccharides are called as glycoproteins and proteoglycans. Glycoproteins are formed by the increased composition of proteins in them, followed by less composition of carbohydrates. Example will be membrane proteins. Then proteoglycans are found to be attached to a specific type of polysaccharide called as glycosaminoglycan. Then third classification, you call them as mucoproteins. These mucoproteins are characterized by specific proteins which are attached by carbohydrate called as N-acetyl galactosamine. Now coming to the next classification, which you call them as lectins. So lectins are very important class of proteins which are called as glycan binding proteins, where they bind to specific oligosaccharides on the cell surface. So these lectins are very important. They act as ligands and they are a class of glycan binding proteins. So these proteins, they bind to the cell membrane carbohydrates with high affinity and high specificity. And these lectins, they are found in all organisms. They are also found in plants, animals, bacteria and viruses. Now if you see, these lectins, they have very important function where they are involved in cell-to-cell recognition and cell-to-cell signaling and cell adhesion. So all of you know what is cell-to-cell recognition. So from one cell to the other, the cells have to recognize the cell membrane receptors which are present. Coming to the cell signaling, certain signaling molecules will react which will identify the reactions between the two cells. Cell adhesion, the cell adhesion molecules which are present on the cell membrane help in the reactions of all the metabolic processes. Then coming to lectins, they are identified as two different forms of lectins, mainly plant lectins, animal lectins. Plant lectins are found in the wheat, Conocavalin A and in resin. Animal lectins are classified as selectins. Now if you see selectins, they are integral proteins of mammalian cell membrane and they play an important role in cell-to-cell recognition and binding. So there are other forms of lectins which are seen as bacterial lectins and viral lectins. So bacterial lectins, example, if you have seen, the common one is cholera toxin, and viral lectins which you have seen as the important one which you call them as influenza virus or heme agglutinin.